Hi everyone, welcome to my channel Swarna Ma'am's Biology. In this video, I will be explaining you about the physiology of digestion in easy and simple way. Let us start. In our last video, we have seen the process of digestion and different components which are involved in the digestive system. And even we have discussed about the different nutrients which are necessary for the digestion. So here, in this video, we will be discussing about the different reactions which are involved in the process of digestion, which we will be calling it as physiology of digestion. Okay, so now when the food is taken inside the mouth, it enters into the buccal cavity. So in the buccal cavity, the food is mixed with saliva. Along with that, there is chewing or mastication of the food also takes place. So after chewing or mastication of food, it is mixing with saliva. During the chewing or mastication of the food, after mixing with saliva, the food is slowly swallowed through the esophagus. So the swallowing of the food is also called as degurgitation of the food. So swallowing of the food or degurgitation of the food is also taking place. So in the buccal cavity, there is chewing or mastication of the food followed by the swallowing of the food is taking place. So when we see here, there is a small leaf-like structure which is present, which is called as epiglottis, which is immediately closing when the food is entering from the pharynx into the esophagus. So here, when the food passes from the pharynx into the esophagus, what this epiglottis do? It immediately closes the larynx so that the food is entering into the esophagus without flowing into the trachea. Why it is doing so means the pharynx is a common opening for the trachea and esophagus. There is a chance of entering of the food from the buccal cavity into the trachea. So what this epiglottis is doing, so immediately it is closing. This epiglottis is immediately closing the larynx, okay, in such a way that the food is now entering into the esophagus without entering into the trachea. What happens when the food enters into the trachea? It causes choking. Okay. So trachea is the part of the respiratory system. We know that esophagus is a part of digestive system. So the food has to enter into the esophagus, then it has to pass into the stomach, then intestine. It must not enter into the trachea and the lungs. What happens if the food enters into the trachea and lungs? It causes choking. So what this epiglottis is doing, it is preventing the flow of the food into the trachea, allowing the food to pass into the esophagus. So let us define what is epiglottis here. So epiglottis is a small leaf-like structure. Okay, it is a small leaf-like structure which is covering the larynx. It is covering the larynx in such a way that the food is entering into the esophagus without entering into the pharynx. Okay, so this is about epiglottis. So now in the buccal cavity, we know that the food is also mixing with saliva. So saliva, when we see, it contains four four enzymes. So that is mucus, electrolytes, salivary amylase, as well as lysozyme. So saliva contains four substances, that is mucus, electrolytes, salivary amylase, as well as lysozyme. So coming to the mucus, what is the function of mucus? Mucus always helps in the maintaining the lubrication of the food. What is the function of electrolytes? They maintain the pH. Whereas what is the function of salivary amylase? This salivary amylase mainly acts on the carbohydrate, that is also starch. So salivary amylase converts the salt starch into maltose. Okay, salivary amylase, which is present in the saliva, that is in the buccal cavity, is converting the starch into maltose. And what is the function of this lysozyme? It is antimicrobial in action. This lysozyme is antimicrobial in action. Okay, in saliva contains four substances, that is mucus, electrolytes, salivary amylase, and lysozyme. Mucus normally helps in the lubrication of food. Electrolytes maintain the pH balance of the buccal cavity, that is saliva. Salivary amylase mainly contains the salivary amylase is an enzyme which mainly act on the carbohydrates which convert the starch into maltose, and lysozyme is antimicrobial in action. So this masticated food, when the food is mixed with saliva, it is masticated. That food we call it as bolus. So here we came across the another word, that is bolus. What is bolus? This food which is mixed with saliva, which is where the starch is converted into more maltose. That food we call it as bolus. Okay, which is called as bolus. Now after that, what happens? That is salivary amylase contains mainly that is salivary amylase mainly act on starch, where the starch is converted into maltose, and the main pH which is necessary maintenance of the pH is 6.8. The pH which is necessary for the maintaining of this reaction is 6.8. Okay, so now, so here, if you see, the saliva contains what? Salivary amylase, which acts on starch, and lysozyme is also present in the that is saliva. So the lysozyme is antimicrobial in action. So that is about saliva. So now, after that, the food after mastication, that is bolus, masticated food, that is bolus, it is now entering into the stomach through the esophagus. Okay, in the stomach, it contains gastric glands. So how many types of gastric glands are present? Three types of the gastric glands are present. So what are they? They are neck cells, peptic or chief cells, parietal or oxentic cells. Okay, three types of the cells are present in the gastric glands. So those are neck cells, peptic cells, parietal or oxentic cells. So now let us see what they produce. So these neck cells, so the neck cells or a mucus cells produces what is called as mucus. So the neck cells produces what is called as mucus. So coming to the peptic or chief cells, so they produce what is called as pepsinogen. Okay, these peptic cells produces what is called as pepsinogen. And the parietal or oxentric cell, they produce HCl. Along with HCl, they also produce an intrinsic factor which is mainly necessary for the absorption of vitamin B12. So here in the stomach, there are gastric glands. There are three types of the cells which are present in the gastric glands. They are mucus or the neck cells, which produces mucus, peptic or the chief cells, which produces pepsinogen, parietal or oxentric cells, which produces HCl. Along with that, an intrinsic factor is also produced, which is mainly necessary for the absorption of vitamin B12. So this is about stomach. So here we have seen that pepsinogen is produced, but this pepsinogen which is produced is inactive. This pepsinogen is inactive enzyme. So this pepsinogen has to act on proteins, but it can't act. It has to be converted into active pepsin. So how it will be converted? So this pepsinogen is inactive. It has to be converted into active pepsin. Okay, so how it will be converted? With the help of HCl. From where this HCl is produced? From the oxentic cells. So this HCl which is produced from the oxentic cells is converting this pepsinogen into active pepsin. So this pepsin is now ready for action on proteins. Okay, so in gastric glands, there are three types of the cells. That is, neck cells produces mucus, peptic or chief cells produces pepsinogen, parietal or oxentric cell produces HCl as well as intrinsic factor. This HCl converts the inactive pepsinogen into active pepsin. Now this pepsin acts on proteins. Okay, pepsin is a proteolytic enzyme. It acts on proteins. Now in the stomach, the food is stored for about 4 to 5 hours. So that semi-digested food, we call it as chyme. So here there is another word, what is called as chyme. Chyme is a semi-digested food where the proteins are partially digested, where the proteins are converted into proteoses and peptones with the help of pepsin, right? So here in the before slide, we have seen that pepsinogen is converted into active pepsin. Yes, with the help of HCl. This pepsin will be acting on proteins. The pepsin will be acting on proteins and is converted into proteoses and peptones. Okay, pepsin is acting on proteins and is converted into proteoses and peptones. These are partial digestion of proteins is taking place in the stomach. And in the stomach, if you see, only proteins are digested. That also partial digestion is taking place. Okay. So this is about the digestion in the stomach. So here also we will see. So in the stomach, what is there? Pepsin is produced from the pepsinogen, and this pepsin is acting on the proteins, where the proteins are converted into proteoses and peptones. Okay. So in the stomach, there are other substances like mucus and HCl. So mucus, we know it is necessary for the lubrication, whereas HCl, what is the function of HCl? It is necessary for the conversion of pepsinogen into active pepsin, and HCl is also antimicrobial in action. It is antimicrobial in action. It is an acid. It maintains a pH so that it is antimicrobial in action. Okay. The HCl is mainly responsible for conversion of pepsinogen into pepsin, and as well as it is antimicrobial in action. So there is about um, that is this is about the HCl as well as mucus as well as pepsin. And there is also another proteolytic enzyme which is called as renin. So this renin is also a proteolytic enzyme, but it is present only in infants. Okay. So renin is a proteolytic enzyme which is mainly necessary for the digestion of milk protein, which is called as casein. And it is also present in some herbivorous animal which eats grass. And renin is a enzyme which is present in the infants which digest the milk protein, which is called as casein. Okay. So here renin is also activated, and it will be acting on the milk proteins. Okay. Now let us take a small concept test here. So the question is, the gastric juice contain pepsin, lipase, and renin, trypsin, lipase, and renin, trypsin, pepsin, lipase, trypsin, lipase, trypsin, pepsin, and renin. So here we can easily answer this. Why? Because trypsin is a proteolytic enzyme which is released by the pancreas. That you have to remember. Wherever trypsin comes, you have to remember that trypsin will be released by the pancreas. Pepsin is a proteolytic enzyme. It is released by the gastric glands, whereas trypsin is also a proteolytic enzyme. It is released by the pancreas. So there is a difference between pepsin and trypsin. Okay. Both are proteolytic enzymes, but trypsin is released by the pancreas, whereas pepsin is released by the stomach. So gastric juice, we can easily answer here. Gastric juice contain, that is pepsin, lipase, and renin. Okay, we have to avoid those three, B, C, D, we have to avoid. So the correct answer is pepsin, lipase, and renin. Okay. So again, you remember trypsin means it is released by the pancreas, whereas pepsin will be released by the stomach. That is gastric glands. Okay. So now we have seen in the stomach what is happening. Mainly the protein digestion is happening. Protein digestion is also not completely happening. Only the proteins are partially digested into proteoses and peptones. So now those proteoses and peptones and other proteins and other substances, everything will be now passing into the duodenum. Now in the duodenum, that is the small intestine. So it will be acted, that chyme will be acted upon by the bile juice, pancreatic juice, as well as intestinal juice. So now in the small intestine, that is in the duodenum, bile, pancreatic juice, as well as intestinal juice, all these juices will be acting on the chyme. That is a partially digested food. We are calling it as chyme. So now we will see the action of bile juice. So when we see the action of bile juice, yes, bile juice as well as pancreatic juice are released into the small intestine by the hepatopancreatic duct. Okay. So when we see the action of bile juice, so bile doesn't contain any enzymes, but bile contains bile pigments as well as bile salts. So what are the two bile pigments? They are bilirubin as well as biliverdin and bile salts. The bile salts are sodium glucocolate and sodium taurocolate, as well as potassium glucocolate and potassium Taurocolate. In simple, what we can say, sodium and potassium glucocolate and taurocolates. So bile juice contain bile pigments, that is bilirubin as well as bilirubirin, and bile salts, that is sodium and potassium glucocolate and taurocolate, and cholesterol. But bile doesn't contain any enzyme. So what these bile pigments and bile salts do? They help in emulsification of fats. They help in emulsification of fats. So what is meant by emulsification? So what is meant by emulsification? It is large breakdown of large fat molecule into small small fat droplets. So breakdown of large fat molecule into small fat droplets, we call it as emulsification. So the bile salts as well as bile pigments help in the emulsification of the fats, where a large fat molecule is converted into small fat droplets. Okay. So now coming to the pancreatic juice. In the pancreatic juice, it also contains several enzymes like trypsinogen, chymotrypsinogen, procarboxypeptidases, amylases, prolipases, and pronucleases. So this pancreatic juice contains so many enzymes, that is trypsinogen, chymotrypsinogen, procarboxypeptidases, amylases, prolipases, pronucleases. But all these are inactive enzymes. They can't directly act. So they have to be converted into active enzyme. So when the pancreatic juice is releasing the enzymes, all these inactive enzymes also are released. They have to be converted into active enzymes. So now these enzymes have to be converted into active enzyme. So trypsin, which is a inactive trypsinogen, also has to be converted into active trypsin. Similarly, all the other enzymes. So we will see the different reactions here. So the enterokinase is the enzyme which convert the inactive trypsinogen into active trypsin. So enterokinase will be converting inactive trypsinogen into active trypsin. Now this trypsin, which is formed now, act on chymotrypsinogen into chymotrypsin. Okay. Similarly, there are other substances, that is procarboxypeptidases will also be converted into carboxypeptidase. Prolipases will be converted into lipases. Pronucleases will be converted into nucleases. Now they have become active. That is, first enterokinase is converting trypsinogen into active trypsin. Then chymotrypsinogen will be converted into chymotrypsin with the help of that trypsin. Then procarboxypeptidases are converted into carboxypeptidases, prolipases are converted into lipases, pronucleases are converted into nucleases. Now all these, that is trypsin, chymotrypsin, carboxypeptidase, lipases, and nucleases, all these are active enzymes. They can act on respective substrates. Okay. Now coming to the intestinal juice. So intestinal juice, we know it is called as succus centricus. So in intestinal juice, mucus also is there, which is produced by the goblet cells. So in the intestinal juice, there are present disaccharides as well as dipeptidases, okay, and even lipases. So these intestinal juice, which contain disaccharides as well as dipeptidases, lipases, as well as nucleosidases, all these are helpful for the further reactions of digestion, that is, which happens in the small intestine. So along with that, this intestinal juice also contains mucus as well as bicarbonates, which also released from the pancreas. So now we will see all the reactions of which are released from the pancreas, and all the reactions of the enzymes which are released from the intestinal juice, which are helpful in completing the process of digestion. So here the mucus bicarbonates which are released from the pancreas along with the Brenner's glands, that is intestinal gland, protects even the intestinal mucosa, that is lining of the mucosa, also maintain the alkaline pH in the intestine. Okay. So it also helps in the maintenance of alkalinity in the intestine. So now coming to the intestinal digestion that is happening in the intestine, that is digestion which is happening in the intestine. So here we know proteins, proteoses, and peptones. are released from the stomach after entering into the intestine. They will be in the form of proteins, proteoses, and peptones. They are converted into dipeptidases with the help of trypsinogen, chymotrypsinogen, and carboxypeptidases. Okay. Then polysaccharides are converted into disaccharides with the help of amylase, and fats are converted into diglycerides with the help of lipases, as well as it is also that glycerides later will be converted into monoglycerides, and nucleic acids will be converted into nucleotides. Nucleotides will be converted into nucleosides with the help of the enzyme which is called as nucleases. Okay, so first trypsin, again I am repeating, trypsinogen, chymotrypsinogen, as well as carboxypeptidases, what they are doing, they are converting proteins, proteoses, and peptones into dipeptidases. And amylase is acting on the polysaccharides and is converted into disaccharides. Then lipases are acting on the fats and is converted into diglycerides. Later, they are converted into monoglycerides. Then nucleases are acting on the nucleic acids and are converted into nucleotides, which are later converted into nucleosides. These are also not complete products of digestion. They have to be further digested. Now again, these dipeptidases acts on dipeptides and converted into monopeptides and amino acids. So dipeptidases are acting on the dipeptides and are converting into amino acid. Now disaccharides are there. They are in the form of maltose, sucrose, and lactose. Okay. So those are disaccharides, means two sugar molecules. We call it as disaccharide. So they have to be converted into monosaccharides. Okay. So now what are those disaccharides? Maltose, lactose, and sucrose. This maltose is a disaccharide which is having two glucose molecules. So this maltose is acted upon by the enzyme maltase and is converted into glucose and glucose. Okay. And lactose is also a disaccharide which is made up of glucose and galactose. Okay. So when lactase enzyme acts on lactose, it will be converted into glucose and galactose. Then sucrase is an enzyme which act on sucrose and again converts into glucose and fructose. Why? Because sucrose is a disaccharide which contain glucose and fructose. So sucrase, which is a disaccharide saccharide enzyme, it acts on sucrose and is converted into two monosaccharide, that is glucose and fructose. So these are the end product of carbohydrate digestion and protein digestion. So what is the main products of nucleic acid digestion? We know that nucleic acids are converted into nucleotides. Now they are again acted upon by the nucleases and is converted into nucleosides, and these nucleosides are again converted into sugar and base. Yes. Okay. So this is about the nucleic acid digestion. And di and monoglyceride, that is about the fat digestion. Di and monoglycerides will be converted into fatty acids and glycerol with the help of lipases. So in the these lipases are acting on the di and monoglycerides and is converted into fatty acids and glycerol. So all these, that is monosaccharides, that is glucose, fructose, those are monosaccharides, as well as fatty acids, glycerol, then amino acids, all these are the end products of digestion. So here we have come across so many reactions during the physiology of digestion. Okay, so I will again write all the reactions, I will summarize. Okay, so now first I will revise everything so that there will be a little confusion up to now. So I will revise so that the confusion will decrease. So first we know that in the mouth, that is in the buccal cavity, the carbohydrate, up to that it is clear, I think. So in the buccal cavity, what it is happening in the buccal cavity? The food is mixing with saliva, and salivary amylase is there. Yes, salivary amylase, what it is doing? Salivary amylase is acting upon the starch and it is converting into maltose. Yes, it is converting into maltose. So this is happening in the buccal cavity. So coming into the stomach, into this in the stomach, what are the reactions which are happening? So mainly stomach, we will be coming across pepsinogen. So in the stomach, We know pepsinogen is an inactive enzyme which is released by the peptic cells of the gastric gland. Yes, but this pepsinogen is an inactive enzyme. It has to get activated. It will be activated into active pepsin with the help of HCl. We know that the HCl is released by the oxyntic and oxyntic cells. So this HCl, what it is doing, it is activating pepsinogen into pepsin. So this pepsin is a proteolytic enzyme. So what it is doing, it is acting on proteins. Proteins are converted into proteoses and peptones, okay, by the action of pepsin. So this part is taking place in the stomach. So in the stomach, only proteolytic digestion is taking place, that also not complete digestion. So this partial digested food, we are calling it as chyme. Now this chyme is passing into the intestine, that is duodenum. In the duodenum, we know three types of the juices are opening into the duodenum. That is pancreatic juice, then bile, as well as intestinal juice. Now, all these juices contain several enzymes which will be acting on that chyme, which contains proteoses, peptones. Along with that, there are other substances which are not still digested. Now, coming to the yeast juice. So, we know that bile juice contains bile salts as well as bile pigments, which acts on fats, where it is converting the large fat droplets into small globules, which we are calling it as emulsification of fats, but not digestion of fats are not taking place. Now, coming to the pancreatic juice. Pancreatic juice contains trypsinogen, chymotrypsinogen, then procarboxypeptidase, then prolipase, pronuclease. All these are inactive enzymes. They have to be activated. Now, enterokinase. So now what happens? This enterokinase, so enterokinase is enzyme which converts this trypsinogen into active trypsin. So it is enterokinase. Okay. Now this trypsin again act on this inactive chymotrypsinogen and converts into chymotrypsin. Okay. This procarboxypeptidase also will be converted into active carboxypeptidase. Okay. Prolipase is also converted into lipase, and nuclease, pronuclease is also converted into active nuclease. Now these pepsin, chymotrypsin, carboxypeptidase, lipase, as well as nuclease are now active enzyme. They are now ready to act on following substrates. Okay. Now coming to the how they are acting. Okay. So now let us discuss how they will be acting. So now coming to the active enzymes, the first trypsin. Now trypsin, procarboxypeptidase, as well as carboxypeptidase, all these will be acting on what are called as proteins. Okay, now let us write the reactions related to that. So now we have active enzymes like trypsin, yes, carboxypeptidase, then amylase, nucleases, okay, and lipases. Now they are now ready for action. Yes. So now proteoses and peptones, these are the products of digestion from the stomach. Yes. Along with some proteins. They will be acted upon by trypsin, chymotrypsin, as well as carboxypeptidase. And these are converted into dipeptides. Okay. Now these dipeptides will be converted into monopeptides as well as amino acids by dipeptidases. Okay, so first proteoses and peptones, which are the products of digestion from the stomach, are acted upon by the trypsin, chymotrypsin, and carboxypeptidases and are converted into dipeptides. Dipeptides are again converted into last monopeptides and amino acids. So these are the final products of digestion of proteins. Okay, now fats. So when we see lipases will be acting upon fats and is converted into diglycerides. And again these diglycerides will be converted into monoglycerides. At last, they will be converted into fatty acids and glycerol. These fatty acids and glycerol are the end products of digestion of fats. Now coming to the carbohydrate digestion. So we know that is a polysaccharide will be digested into disaccharides. So disaccharides, what are the different forms of disaccharides? They are maltose, sucrose, and lactose. These are disaccharides. Now, this polysaccharides is converted into disaccharides, okay, by amylases. And this disaccharide will be acted upon by disaccharase. Okay, ASS means it is an enzyme which is acting on a particular substrate. Okay, this disaccharide is acted upon by disaccharase and it is converted into monosaccharides. Okay, so first it is converted into monosaccharides. So example, maltose is one of the disaccharide. So this maltose is acted upon by maltase. It is acted upon by maltase. AS means enzyme. Maltose is a, we know it is a disaccharide molecule. This maltose is acted upon by maltase and is converted into two glucose molecules. As maltose is a disaccharide containing two glucose molecules, so it is acted upon by the maltase where the maltose is split up into two glucose molecules. Then sucrose is acted upon by the sucrase and is converted into glucose and fructose. Why? Because sucrose is a disaccharide which contains glucose and fructose components. So when sucrase is acting on sucrose, sucrose will be broken down into two monosaccharide, that is glucose as well as fructose. Now coming to the lactose. Lactose is acted upon by lactase and is again converted into glucose and galactose. What are these? Means these are also monosaccharides. Glucose and galactose are a monosaccharide and lactose is a disaccharide. So at last, what we have to see, all the disaccharides are also converted into monosaccharide. So here we have seen the end product of that is a protein digestion. What are the end products of protein digestion? It is amino acids, whereas the end product of that is a fat digestion, it is fatty acids and glycerol, and product of carbohydrate digestion, that is monosaccharides. Yes, monosaccharides. Now coming to the nucleic acids. Coming to the nucleic acids. These nucleic acids are also acted upon by the nucleases. So nucleic acids are acted upon by the nucleases, where these nucleic acids are converted into nucleotides. They are converted into nucleotides. Later, these nucleotides are converted to what are called as nucleosides. Later, these nucleosides are converted into base plus sugar. So this is the end product of digestion of nucleic acid. So at last, what are the end products of digestion? They are amino acids for proteins. Yes. Then fatty acids and glycerol for fats. Yes. Nucleic acids at last, it is base and sugar. And carbohydrate digestion, it is monosaccharides like glucose, fructose, and galactose. These are monosaccharides. Yes, disaccharides are converted into monosaccharides. So these amino acids, fatty acids and glycerol, base and sugar molecule, monosaccharides, all these are end products of digestion. So all these reactions involved are are involved in the process of digestion, which we have discussed in the physiology of digestion. Okay, so this is about the physiology of digestion. Okay, at last we have discussed that the end products of digestion are amino acids, fatty acids and glycerol, base and sugar molecule for the nucleic acids, as well as monosaccharides for the that is polysaccharide. Polysaccharide is converted into monosaccharide. So this is about the reactions which are involved in the physiology of digestion. Hope you understood this topic. So this is about the physiology of digestion. Hope you understood this topic. If you like this video, please give a like, share and subscribe my channel, Swarna Ma'am's Biology. Okay, thank you. Meet you in the next video, where we will be discussing about the absorption as well as assimilation. Okay, thank you. Bye.